Hi everyone. Today we will talk about viral vector vaccines, non-replicating viral vector vaccines. Vector is a vehicle used to transfer the genetic material into the target cell. Several different viruses have been used as vectors, including influenza virus, vesicular stomatitis virus, measles virus, and adenovirus, which causes the common cold. Viral vector-based vaccines differ from most conventional vaccines because they don't actually contain antigen, but rather they use the body's own cell to produce them. They do this by using a modified virus, which called the vector, to deliver genetic code for antigen. So the vector serves as a delivery system that carries the genetic code to make a specific protein. In the case of SARS-CoV-2, it's the spike protein. Vectors, they are derived from viruses because viruses are efficient in getting into the cell. But for safety purposes, all of the viral genes are removed and the vector is modified to only deliver therapeutic gene. So they uses the capsid of the virus. So there are two main types of viral vector-based vaccines. So first is the non-replicating vector vaccines. They are unable to make new viral particles. They only produce the vaccine antigen. So they are non-integrating, which means that they do not insert itself into the cell's genome. So if it is taken up with the cell that divide, the therapeutic gene won't be copied with each cell division. That's why they are used in non-dividing target cell. So next is the replicating vector vaccines also produce new viral particles in the cell they infect, which then go on to infect new cells that will also make the vaccine antigen. So they integrate into the genome of the target cell. That's why they are used in dividing cells. So based on the picture, an example of non-replicating viral vectors are the adenovirus and adeno-associated virus, while the lentivirus and retrovirus is a replicating viral vectors. So the vaccine for COVID-19 are non-replicating, which require higher doses but are safer than replicating viral vectors. So again, replicating viral vectors, they produce new viral vector particles in cells they enter, while the non-replicating, they don't produce new viral vector particles in cells that they enter. So different viruses can be used as viral vectors in these vaccines. The COVID-19 viral vector vaccine candidates use a range of different viral vectors to deliver their genetic cargo. So the companies that uses human adenovirus as vectors, so we have the Gamaleya Research Institute that uses the adenovirus type 5 and adenovirus type 26. The Johnson& Johnson that uses the adenovirus type 26 and the CanSino Biologics that uses the adenovirus type 5. While the while there are also companies that uses primate adenovirus as vectors. So we have the Oxford AstraZeneca that uses adenovirus in chimpanzee and the Raytera that uses adenovirus in gorilla. So as seen on the picture, we have the vaccine that contains a modified virus, the adenovirus. So the adenovirus has been modified so it is now harmless because the disease-causing genes are removed and replaced with therapeutic gene that contains genetic material to make spike protein. So once injected into the body, these vaccine viruses begin infecting our cells and inserting their genetic material, including the antigen gene, into the cell's nuclei. So human cells manufacture the antigen as if it were one of their own proteins, and this is presented on their surface alongside many other proteins. So when the immune cells detect the foreign antigen, they mount an immune response against it. This response includes antibody producing B cells as well as T cells, which seek out and destroys infected cells. So advantages of viral vector vaccines. So viral vectors cannot cause infection with COVID-19 or with the virus used as the vaccine vector. The genetic material delivered by the viral vector does not integrate into a person's DNA. And it has the advantage of triggering a strong cellular immune response by T cells as well as the production of antibodies by B cells. So the disadvantages. So previous exposure to the virus vector can develop an immune response against it, reducing the effectiveness of the vaccine. So that's why delivering the second dose is a challenge because of the anti-vector immunity, but this can be resolved if the second dose will going to use a different virus vector. So a major bottleneck for viral vector vaccine production is scalability. Traditionally, viral vectors are grown in cells that are attached to a substrate rather than in free-floating cells. This is difficult to do on a large scale. That's why suspension cell lines are being developed to enable the viral vectors to be grown in large bioreactors. And then assembling the vector vaccine is also a complex process involving multiple steps and components, each of which increases the risk of contamination. So extensive testing is therefore required after every step, causing increasing cost. So that's all for the viral vector, non-replicating viral vector vaccine. So let's get vaccinated and end this pandemic. Thank you.